All right, hello, and thank you all for joining us during the Lytham Partners Investor Select Conference. My name is Robert Blum, Managing Partner of Lytham Partners. During this webcast, we welcome Shuttle Pharmaceuticals, ticker symbol of SHPH on the NASDAQ, and their CEO, Dr. Anatoly Truchillo. Today, Dr. Truchillo will run through the slide presentation. Before we begin, I just want to remind everyone that if you've not already signed up for a one-on-one meeting and would like to do so, please send me an email at Blum, B-L-U-M, at lythampartners.com, or you can visit our website, lythampartners.com/virtual. Click on the one-on-one meeting request button there, and we will look to get you taken care of. So, Dr. Truchillo, the floor is yours. Please begin.
Good afternoon, Robert, and thank you for the opportunity to present our company's research to improve outcomes for cancer patients undergoing radiation therapy. I will be presenting forward-looking statements. We are a public company, and there's abundant disclosure information that has been submitted to the SEC. My background is summarized here as an academic clinician, researcher, and entrepreneur. I have served as department chairman, hospital medical director, and interim cancer center director at Georgetown University for most of my career. As a radiation oncologist, I have personally treated more than 5,000 cancer patients, and my research has been supported by more than $20 million in grant support from government and industry sources. Shuttle Pharma is a clinical stage biopharma developing ropidoxyuridine as a drug to sensitize cancers to the effects of radiation therapy, and HDAC6 selective inhibitor SP225 to stimulate the innate immune system to target cancers in the immediate post-radiation period. Our mission is to improve the lives of cancer patients. These treatments will make the radiation more effective and to enhance immune efforts to help control tumors. The fundamental problem in radiation oncology is described in this slide. Currently, we have outstanding technology to deliver radiation therapy. We can deliver any dose to any location in the body. The problem is that there are normal tissues that are located in very close proximity or adjacent or involving the cancerous tumor. So in order to treat these, we'd like to get to a therapeutic dose, which is generally a high dose of radiation, but we're limited if we have normal tissues that are sensitive. Therefore, the approach should be to make the cancer cells more sensitive to radiation and to recruit the immune system to attack the cancer cells. And this is the main target of our radiation sensitizers and innate immune stimulators. Our lead sensitizer and lead HDAC6 inhibitor are summarized on this slide. We are developing ropidoxyuridine to treat brain tumors, and we are developing SP225 for potential use in myeloma, lung cancer, and breast cancer. Currently, there are a number of drugs that are used as radiation sensitizers by clinicians. These drugs are used off-label. They were not approved for sensitization, but they're used because they do have some effects. With the exception of cetuximab, which does have FDA approval as a radiation sensitizer for treating head and neck cancers, these drugs are off-label, and they're listed here as a sample of the types of cancers that are treated and side effects and the previously obtained revenue numbers. So the market opportunity is substantial. If we consider patients who undergo a course of radiation therapy for local or local regional control of disease, these would all be potential candidates for a sensitizer. I've summarized here the market opportunity, which is 1.8 million patients with cancer. This is of which about half are treated with radiation, and of those, about half are treated with curative intent. So we're looking at about 450,000 patients a year that fall into this category. We summarize our clinical pipeline on the slide. Ropidoxuridine is advancing to phase 2 clinical trials. Our focus will be on brain tumors. We are also collaborating with other investigators to develop the drug for sarcomas. We have sufficient resources to advance SP225 through IND enabling and into phase 1 clinical trials. So the other inhibitors will be developed as we identify additional resources. Ropidoxuridine is an orally available prodrug of IUDR. IUDR is an active compound that is known to be an extremely powerful radiation sensitizer. The problem with IUDR in the past has been that it has a short half-life and it has to be delivered by intravenous infusion. So we have the prodrug, which is the ropidoxuridine, gets metabolized to IUDR, and critically, it is taken orally as a pill. Here we summarize some of the work that's been done with this drug, and there has been a clinical trial, both a phase 0 and a phase 1, and supports the tolerability of this drug in combination with radiation therapy and the bioavailability in the bloodstream. The mechanisms are basically the drug is metabolized into IUDR, the prodrug. The IUDR is incorporated into the DNA, and when radiation exposure is provided, there are increased DNA strand breaks due to the generation of additional free radicals. The drug is not effective as a radiation, as a single agent. It's only effective when given in combination with radiation therapy. The clinical trial established a maximum tolerated dose of 1,200 milligrams daily, given daily for up to 28 days. The clinical trial also showed that there were 4 partial responses. There were 9 stable disease evaluations, and only 1 patient progressed while on this treatment. Our development of ropidoxuridine has been targeted toward manufacture and preparation for advancing the phase 2 clinical trial. We have engaged TCG GreenChem to manufacture the drug, and we are looking to have this API manufactured by this summer. The engagement of Theradex is to assist us in the management of the phase 2 clinical trial and to also assist us with the regulatory functions with the FDA. Theradex was previously the company that performed the phase 1 clinical trial. So the next steps in the development of these drugs, we have 2 for ropidoxuridine, the development of the drug itself through phase 2, and we'd like to develop it for the treatment of brain tumors. We have received FDA approval for advancing this under the orphan drug designation category for brain tumors. And we have also developed some new formulations to enhance the availability of the drug for colorectal cancer. That particular formulation is an improvement in the bioavailability. We shift over to the HDAC inhibitor platform, which is currently in preclinical development in our company. This shows you the actual development, how we synthesize these drugs. And we basically changed the HDAC selective domain by applying domains, motifs that allow us to be selective and in terms of binding to the HDAC molecule enzyme for inhibition. The HDAC6 molecule is particularly targeted for us because it's involved in significant effects of the immune system. By inhibiting HDAC6, you prolong the immune system activation against tumors. And this is a critical part of the treatment of these cancers. We have synthesized these drugs. We have screened them all in our laboratories, and we own all of the intellectual property for these molecules. Here I show a small effort that was performed with our collaborator, Dr. Alejandro Villagra at George Washington University under a material transfer agreement. And basically what we show is that by combining the drug with radiation, the antigens on the surface of the cancers are increased, and they become a better target for the immune system. The graph on the right shows that when this is performed and we treat this with a mouse model of melanoma, we show tumor inhibition, and this allows us to use only the immune system to attack these cancers. So the point being that if a patient gets radiation, there are a number of antigens that are brought to the surface of the cancer, and those antigens become the target of the immune system. So we can now target both the immune system to this, and we can also sensitize the cancers to radiation. And this provides a dual approach to the treatment of cancers. Our intellectual portfolio is summarized here. The ropiduxuridine is, we've applied for formulation, and we're also developing some manufacturing patents. The formulation patent was actually approved last week by we received notice of allowance from the U.S. Provisional patent submission. And the dual functional isoform selective HDAC inhibitor platform is listed here. We have, at this point in time, we have 3 of our patents have been approved, and there are several currently still under review and are pending approval. In summary, the development strategy for radiation sensitizers, we have 2 major priorities. The first one is to advance ropiduxuridine in combination with radiation therapy to treat brain tumors. The focus of our work is initially to manufacture the drug, develop the phase 2 clinical trial, and to develop the treatment under orphan designation. The second priority is the development of the HDAC6 selective inhibitor, SP225, for activation of the immune system after radiation therapy. For that, we have a process established for manufacturing a smaller amount of drug to perform the IND-enabling studies, and then to advance to IND and a phase 1 clinical trial. The financial overview as of 11/30/2022 is summarized here. We have taken the company public, and we have raised, at that point, $10 million. We have subsequent secondary raise of additional $4 million for the SP225 project. This is our leadership team. The team's experienced, and we have worked together for many years. I function as the CEO and chairman of the board. My son serves as the president and chief operating officer, managing all the business items associated with the company. Michael Vanderhook is a former administrative director of Lombardi Comprehensive Cancer Center, and I've worked with Michael for approximately 15 years. Same with Dr. Rich. He's our medical director. He also trained with me and has a strong background in the development of sensitizers. Mary Young develops the laboratory assays and laboratory testing. And the chair of our scientific advisory committee is Theodore Phillips. He's a member of the National Academy of Sciences. Thank you for your attention, and Robert, thank you for this opportunity to present the company.
Fantastic. Thank you so much, Dr. Chillo. Really appreciate your time and the presentation here today. I guess I'll just remind everyone, if you have not already signed up for a one-on-one meeting and would like to do so, again, please send me an email, Blum, B-L-U-M, @lythampartners.com, or again, visit the website, lythampartners.com/virtual, click on the one-on-one meeting request button, and we'll look to get you taken care of. So we hope you all enjoy the conference. Have a great rest of your day. Thank you so much, Dr. Chillo.
Okay.